Hello everyone, welcome back to my course on microbial biotechnology. We're in module 6, where today we will be discussing about microorganisms in pharmaceutical industry. So we have divided this lecture into 5 sections. Section 1 will discuss about microorganisms overall in the pharmaceutical industry. Then number 2, drugs from microorganisms, and number 3, biotransformation of steroids. Number 4, live organisms as therapeutic agents, and section 5 will deal with other therapeutic agents like vaccines, nutrient supplements, and probiotics. So let us have an overview of pharmaceuticals and microorganisms. The profound impact of antibiotics on human health over the past 6 decades has been substantial, and these stand as one of the most critical groups of compounds synthesized by industrial microorganisms. Other significant healthcare products obtained from microbial fermentations or biotransformations include alkaloids, steroids, toxins, vaccines, and we will have a detailed discussion of these in module 10. And others such as vitamins, specific enzymes, and live microbial cell preparations which are used as probiotics. Advancements in genetic engineering have enabled microorganisms to produce an extensive array of mammalian proteins and peptides with diverse therapeutic properties, apart from the traditional uses of microorganisms to produce antibiotics and metabolites. Among the medically significant ones, for example, insulin, interferons, human growth hormone, and monoclonal antibodies have established markets. And beyond these, therapeutic agents that alleviate or treat diseases many diagnostic products have stemmed out from application of microorganisms. What role microorganisms play as pharmaceutical or therapeutic agents? They play a crucial role in the pharmaceutical industry across various aspects. For example, in the case of drug development, microorganisms are used in the development of antibiotics, antifungals, and other medications. Example, penicillium molds are used for producing penicillin, streptomyces for streptomycin, and various fungi and bacteria are used for producing other antibiotics. They are used for vaccine production. Many vaccines, including those against measles, mumps, rubella, and hepatitis, are made using vector to kill forms of microorganisms or virus. These stimulate the immune system to produce antibodies without causing disease. Then there are the biological therapeutics or biotherapeutics. Microorganisms are employed in the production of these biotherapeutics like insulin, growth hormones, clotting factors, which are used in treating various conditions. And genetically engineered bacteria and yeast are used to produce these biotherapeutics. Then we have the probiotics. Beneficial organisms like certain strains of bacteria and yeast are used as probiotics to promote gut health. Then enzymes, microbial enzymes are widely used in pharmaceutical processes, aiding in drug synthesis, purification, and formulation as a drug itself. Let us discuss about the screening for potent bioactive compounds. Several natural products have long been utilized globally in treating various human ailments, gaining recent attention for their therapeutic potential. Yet, natural products consisting of numerous chemical elements pose challenges in pinpointing the specific active compounds responsible for their effectiveness. The traditional approach to screen bioactive compounds in natural products involves isolating specific compounds, assessing their pharmacological activity, and studying their pharmacokinetics through effective. This method is time consuming and demanding. Diverse biological screening methods have been used for various targets such as DNA, proteins, and receptors. When biological targets are exposed to natural product extracts, active compounds have the potential to selectively bind to these targets, while unbound compounds are removed readily. Subsequently, the isolated active compounds can be analyzed using chromatographic techniques paired with mass spectrophotometry or other detection systems. So, some of the methods for screening bioactive compounds involve various techniques aimed at identifying the evaluating compounds with potential therapeutic effects. Some of these methods include HTS or high throughput screening. This is a technique used to rapidly test a large number of compounds against specific biological targets, allowing for the identification of potential drug candidates. bioassays. These assays involve using living organisms, tissues, cells or enzymes to test the biological activity of compounds and assess their potential effects on specific biological processes. Chromatography techniques like HPLC, GC separates and analyzes complex mixtures, aiding in isolating and identifying bioactive components. Mass spectrophotometry, MS identifies and quantifies compounds by measuring mass to charge ratios, enabling molecular characterization. Then there we have computational methods like molecular docking, which simulates small molecular target protein interactions predicting binding affinity and efficacy. Then we have cell based assays. These assays assess compounds effects on cellular processes like proliferation, apoptosis and gene expression. Then we have receptor based screening. These evaluates compounds for interactions with specific diseases related receptors or proteins. Then we have enzymatic assays. These test compounds impact on enzyme activity, providing insights into metabolic pathways or disease mechanisms. Let us now move to section 2, where we will discuss about drugs from microorganisms. We get various kinds of therapeutic molecules like antibiotics, anti-tumor drugs, immunosuppressants, enzyme inhibitors, anti-hypersensitives and antivirals utilizing microorganisms. In this picture you can see a cristapen injection, which is basically benzene penicillin sodium. This is used in the form of injections. So, numerous drugs are derived from primary and secondary metabolites in microorganisms. Of over 500,000 bioactive compounds screened, 20 to 25% are microbial in origin, with actinomycetes contributing 45% of these and fungi 38% and unicellular bacteria around 17%. With rising antibiotic resistance, the need for new drugs is increasing, highlighting the importance of microorganisms in producing antibiotics and other medical substances for treating severe ailments. Let us have a brief discussion on the history of microbial drug discovery. The era of microbial drugs began in 1928 with Alexander Fleming's accidental discovery of penicillin. It was isolated in pure form and became a potent antibacterial agent during World War II, with its structure elucidated in 1945 by Fleming, Chain and Flore. And you can see here Sir Alexander Fleming, who was knighted, examining a bacterial culture plate in this figure. Fleming's work led to the isolation of other antibiotics like chloramphenicol and streptomycin. By 1997, 42% of top selling drugs were derived from microbial metabolites. Advances in antimicrobial drugs such as teicoplanin for resistant gram-positive infections and glycyl cyclines for tetracycline resistant bacteria showed promises in combating resistant bacteria. Most antibiotics are secondary metabolites produced by filamentous fungi and bacteria, particularly actinomycetes. Although over 4,000 antibiotics have been identified, only about 50 are consistently used in antimicrobial therapy, with beta-lactams, aminoglycosides and tetracyclines being the most common. Others fail to meet key criteria due to issues like lack of selectivity, toxicity or high production costs. Some antibiotics also serve non-conventional roles. Actinomycin and mitomycin from Streptomyces pusetius and Streptomyces cipitosus respectively act as anti-tumor agents. Some antibiotics are used to manage microbial diseases in crops. Certain antibiotics are used as growth promoters in animal feed. The beta-lactams. More than 100 beta-lactams, predominantly penicillins and cephalosporins, have obtained approval for human usage, constituting over half of the global antibiotic production. So, in this figure 1, you can see the skeletal formula of the basic structure of penicillin, and in figure 2, the basic structure of cephalosporin antibiotics. And you can see the beta-lactam ring, which is highlighted in red in both the cases. These beta-lactams category stand out for its broad safety margin. These compounds specifically target the synthesis of peptidoglycan, a crucial component of bacterial cell walls, absent in eukaryotic organisms, ensuring a high level of specificity. Their primary action involves inhibiting the cross-linking transpeptidation process, which leads to the formation of incomplete peptidoglycan, significantly compromising the structural integrity of bacterial cell walls. You can refer to module 5, lecture 4, where we have discussed this. Let us now discuss over penicillin in little bit detail. The fundamental structure of penicillins is 6 amino penicillinic acid or 6 APA, which includes a thiazolidine ring combined with a beta lactam ring. So, here you can see the structure of penicillin showing this thiazolidine ring in green and the beta lactam ring in red. The 6 amino position of this structure contains various acyl substituents. The beta lactam thiazolidine framework is produced from L-alpha amino adipate, L-cysteine and L-valine, and is shared among penicillins, cephalosporins and cefamycins. When side chain precursors are not added to the fermentation medium of Penicillium notatum or Penicillium chrysogenum, a mixture of natural penicillins, particularly penicillin G and the more acid resistant penicillin V is obtained from culture filtrates. So, here in this picture we can see in the top penicillin fermentation vessel from the 1940s, and in the bottom we can see a 1957 bioreactor which was used to grow the penicillium mold. Previously penicillin was commercially manufactured using a stationary mat culture, and production was achieved through surface processes including solid state fermentation and surface liquid fermentation. Currently penicillin is produced through suspension culture fermentation, where Penicillium chrysogenum is cultivated under controlled agitation. The process is entirely aerobic and utilizes a fed-batch method to maximize efficiency. So, here you can see the diagrammatic representation of the general scheme for penicillin production. First we go for the preparation of the media. So, there are various ingredients which are put into a tank for mixing, and then these are sterilized, and then these are fed into the fermentation or the fermenter or the bioreactor. Then there is a preparation of the inoculum. We start with a small inoculum, then we go for laboratory culture, and then this is fed into a seed tank for increasing the number. So, that this inoculum is again added to this fermenter. So, we have media and we have inoculum both put into this fermenter. So, the fermentation process takes place. Then after this we go for the downstream processing and recovery of the penicillin. So, this is in brief the process of penicillin fermentation. So, penicillin fermentation uses glucose, lactose and corn starch liquor CSL in the fermentation media. Fermentation lasts for around 6 days, and the fermentation process is usually conducted at 26 degrees centigrade and the pH is maintained at around 6.5. The fermentation occurs in 3 phases. During the active growth phase, the nutrients in the medium are rapidly consumed while maintaining a constant pH. Towards the end of the active growth phase, the nutrients are depleted, leading to release of ammonia due to deamination reactions, causing the pH to rise to 7 to 7.5, an optimal range for penicillin synthesis. The late growth phase is succeeded by the ideo phase, where penicillin synthesis occurs at growth halts due to nutrient depletion. It is essential to prevent the late ideo phase from progressing too far to avoid cell lysis and subsequent penicillin degradation. Penicillin biosynthesis comprises three primary stages: the catalytic step, the oxidative step, and chain exchange. During the catalytic phase, ACV synthetase enzyme facilitates the condensation of cysteine, valine, and alpha amino adipate into ACV tripeptide. This particular molecule is produced by the condensation of these three molecules. The tripeptide ACV then undergoes oxidative ring closure, forming a bicyclic ring facilitated by the isopenicillin N synthase to give isopenicillin N. So, due to this ring closure, the isopenicillin N is produced. The final step involves the substitution of L amino adipate. The conversion of isopenicillin N into penicillin G is achieved by utilizing a two enzyme system comprising acyl coenzyme synthetase and acyl coenzyme racemase. So, this is the final product, the penicillin G coming out as a result of this two enzyme system interaction. Now, apart from this natural penicillin, we have many derivatives of penicillin. Semi-synthetic penicillins are created through a combination of microbial, enzymatic or chemical processes. The key intermediary in this production is 6-APA, essential in the synthesis of semi-synthetic penicillin. For commercial production of 6-APA, hydrolysis of penicillin G and V is carried out using an enzyme like penicillin acylase or amidase. Microbial hydrolysis of natural penicillins occurs optimally at higher temperatures and an alkaline pH. Once obtained, 6-APA is chemically bonded with the desired side chain. Let us now have a discussion on the anti-tumor drugs which are produced by microbial fermentation. Microbial metabolites play a crucial role in cancer therapy, with over 60% of approved small molecule drugs derived from natural products or their modified forms, often sourced from Streptomyces initially screened for tumor cell activity. So, here we can see actinomycin D, a widely used chemotherapeutic drug which is marketed as dactinomycin. The use of microbial metabolites as anti-tumor agents dates back to the discovery of these actinomycin in the 1940s. Since then, numerous compounds with anti-cancer properties have been isolated from natural sources. Notable among these approved drugs include actinomycin D, anthracyclines, then bleomycin, mitomycin C, anthracenones, anidines, Taxol and epothyllones. So, here we can see a list of anti-tumor drugs like mitomycin, anthracyclines, streptozotocin, bleomycins, epothyllones, Taxol, etc. And we can see the various organisms which produces these drugs. For example, mitomycin is produced by Streptomyces cyphatosus, and the mechanism of action is forming covalent bonds across the DNA strands, and this is effective against hypoxic cells in certain neoplasms like gastric, colorectal and lung cancer. Similarly, we have the anthracyclines produced by S. pusatius, and it mostly acts through the intercalation function and inserts in between the adjacent base pair of DNA, effective against lung cancer. Then you have the streptozotocin produced by Streptomyces achromogenes through alkylation, which is toxic to cells by causing damage to DNA and effective against pancreatic islet cancer cells. Then you have the bleomycins produced by Streptomyces verticillus. It induces DNA strand nicks, effective against carcinomas and lymphomas. Then we have epothyllones produced by Sorangium cellulosum. Its mechanism of action includes stabilizing microtubulins and inhibiting P glycoprotein efflux pumps, effective against breast cancer. Then we have caliciamycins, which are produced by micromonospora, and it acts by the cleavage of double stranded DNA, and it is effective against acute myelogenous leukemia. Then we have Taxol or paclitaxel, produced by various organisms like Taxomyces and Nodulisporium. It acts by promoting tubulin polymerization, interfering with normal microtubule breakdown during cell division, and it is effective against refractory ovarian cancer, breast, and advanced forms of Kaposi's sarcoma. Let us discuss about another class of compounds which are obtained from microbes. These are the immunosuppressants. Immunosuppression refers to reducing the immune response to prevent graft or transplant rejection or to treat autoimmune diseases. Several microbial compounds have been identified with the capability to suppress the immune response. So, here you can see the structure of sirolimus, which is marketed as a drug, is a known immunosuppressant. Cyclosporin, derived from the fungus Tolyplochedium inflatum, is a calcineurin inhibitor and has been used since the 1980s. Then there is rapamycin from Streptomyces. It complexes with FK protein 12 to deactivate the mammalian target of rapamycin. Then we have tacrolimus from Streptomyces succubensis. This is also a calcineurin inhibitor, but shows 100 times more potency than cyclosporin. All these actions ultimately affect the secretion of interleukin 2, blocking the activation of B cells and T cells. Another important class of compounds are the enzyme inhibitors, which are obtained from microbes. Enzyme inhibition is a mechanism of disease therapy by blocking certain disease pathways. So, enzyme inhibitors are valuable tools for studying enzyme structure and reaction mechanisms, as well as for applications in medicine and agriculture. Microbes produce various enzyme inhibitors with industrial uses, including clavulanic acid. It is a beta lactamase inhibitor, and statins. These are cholesterol lowering drugs. Other inhibitors target enzymes such as glucosidases, amylases, lipases, proteases, and xanthine oxidases. So, here is a table of various enzyme inhibitors obtained from various microbial sources, and which also shows the particular enzyme that is inhibited by the particular inhibitor, which is used as a drug. And in this column you can find the pharmacological importance of these various enzyme inhibitors which are used as drugs. For example, the inhibitor clavulanic acid produced by Streptomyces clavuligerus, it basically inhibits the beta lactamases. So, its pharmacological importance lies in the fact it is an inhibitor of beta lactamases, helps in overcoming antibiotic resistance against beta lactam group of antibiotics. Then we have other examples like acarbose, which is produced by actinoplanes, and it inhibits alpha glucosidases and sucrase. So, it will have decreased starch breakdown in the intestine, and it is therefore useful in combating diabetes. Then we have amylase inhibitors produced by Streptomyces, and then it inhibits amylase, and therefore it prevents dietary starches from being absorbed by the body, useful in diabetes, obesity, and hyperlipemia. So, these are currently very important lifestyle diseases. Then we have lipstatin, again produced by Streptomyces toxotrichini, and it inhibits the pancreatic lipase, thereby interferes with the gastrointestinal absorption of fat. Then we have certain protease inhibitors like antipain, leupeptin, then chymostatin, produced by various species of Streptomyces, and they target the enzyme protease, and their pharmacological importance lies in the fact in used against emphysema, arthritis, pancreatitis, and also cancer and AIDS. Then we have hydroxyacalone, which is produced by Agrobacterium aurantiacum, and it targets the enzyme xanthine oxidase, and it prevents excessive accumulation of uric acid. So, if you can see here, different enzyme inhibitors can help us address various diseases so common in humans. The next important type of compounds produced with the help of microbes or obtained from microbes are the anti-hypersensitives. Particularly statins are potent anti-hypersensitive drugs which work by reducing cholesterol levels through the inhibition of HMG coenzyme A reductase. So, blocking of these enzyme in the liver activates low density lipoprotein receptors, leading to increased LDL clearance from the bloodstream and decreased blood cholesterol levels. Another class of drugs, the jarajogic acids act as competitive inhibitor of squalene synthase, blocking the binding of farnesyl pyrophosphate. This inhibits the conversion of farnesyl pyrophosphate to squalene, which ultimately leads to a decrease in cholesterol synthesis. This cholesterol lowering action mitigates the risk of cardiovascular disease and prevents stroke. So, here you can see the statin, which is inhibiting the HMG coenzyme reductase, and then here it is the jaragogic acid, which is a competitive inhibitor of the squalene synthase, and thereby it is blocking the binding of farnesyl pyrophosphate and inhibiting the conversion of farnesyl pyrophosphate to squalene, which ultimately leads to a decrease in the cholesterol synthesis. What are the sources of anti-hypersensitives? The first statin, compactin, was initially derived from Penicillium brevicompactum and later from Penicillium citrinum as an antibiotic product. Derivatives of compactin such as lovastatin, an ethylated form, was isolated in the 1970s from Monascus rubra and Aspergillus terreus broths, becoming the first commercially marketed statin approved by the FDA in 1987. Another statin, pravastatin, is produced through distinct biotransformation methods from compactin by Streptomyces carbophilus and Actinomadura species. Jarajogic acids A, B and C were initially identified from distinct sources and unidentifiable sterile fungal culture, Sporomiella intermedia and Leptodontidium elateus near the European city of Zaragoza, Spain, hence the name jaragozoic acids. Now, let us discuss about some of the metabolites which act as antivirals. Since the 1980s, HIV/AIDS has posed a huge risk to human health, especially the vulnerable communities of the society. And recently, with the COVID-19 pandemic, it has become much apparent that viruses pose a huge risk to human health, and as such there is need for potent antiviral compounds. So, this has led to an increase in study of antiviral drugs from various sources, and more than 100 different drugs from microbial sources are now known. Recent studies indicate that natural compounds synthesized by microorganisms such as orasparnon A, neoquinolin A and B, aspulvinone D, M and R exhibit significant in vitro potency against SARS-CoV-2 virus, and here you can see that orasparnon A, which is an anti-SARS-CoV-2 drug obtained from Aspergillus niger. So, these are some of the bioactive compounds which has antiviral activities. You have the metabolite name in this column here, then you have these organism from where these metabolites are obtained, and you can see the name of the virus against which these are used, and here you can see the mechanism of action. Let us now move on to section 3, where we will be discussing about the biotransformation of steroids. We will have first an overview of the structure of steroid and the general scheme for steroid biotransformation, and then finally the industrial scale biotransformation. So, steroids are known as cyclopentanoparhydrophenanthrene. These are natural compounds with various therapeutic properties. In this figure, we can see certain structures of common steroids with a steroid nucleus, then testosterone and progesterone. More than 250 steroids and their derivatives are found in various organisms like insects, plants, vertebrates, fungi. It has been observed that even minor alterations in the molecular structure of steroids can significantly impact their biological activity. Consequently, extensive research has been undertaken to enhance the activity of existing steroid compounds and synthesize novel steroidal compounds with pharmacological efficacy. Steroid biotransformation. Steroid transformation converts initial steroids into drug intermediates, which are then processed into active compounds through simple chemical or microbial methods. Chemical synthesis of steroids often require numerous steps, hazardous reactions, and creates environmental challenges. For example, producing cortisone from deoxycholic acid involves 37 steps under extreme conditions, resulting in costs exceeding $200 per gram. What are the processes involved in the biotransformation? Numerous processes are there, starting from hydroxylation, then dehydrogenation, epoxidation, ring A aromization, side chain degradation, reduction of aldehydes and ketones to alcohol, hydrolysis, esterification, and degradation of steroid nucleus. So, in hydroxylation, there is a substitution of hydroxyl group for hydrogen. In dehydrogenation, there is an introduction of CC double bond on the rings by removal of water. In epoxidation, addition of a single oxygen atom across a CC double bond takes place. In ring aromatization, introduction of aromatic ring A through oxidation happens. In the side chain degradation, selective removal of aliphatic side chain without breakdown of the steroid nucleus happens. In the hydrolysis, addition of water by acetylases to degrade steroidal acetates takes place. In the esterification, it generally involves acetylation, and then here you have various examples, like for example, cortisone to hydrocortisone by Carvularia lunata, and so on. Let us look for the general scheme. Steroid production solely through biotransformation is not generally feasible because of the multiple steps involved, and thus a combination of microbial transformation and chemical reaction is employed. For instance, conversion of stigmasterol from soy to cortisol involves the following steps. The first step is the chemical conversion of sigma sterol to progesterone. and biotransformation of progesterone to yield 11 alpha hydroxy progesterone by Rhizopus nigricans. Then chemical conversion of 11 alpha hydroxy progesterone to cortisol. Similarly, diosgenin from Mexican Barbasco plant can yield cortisol in the following steps. Chemical conversion of diosgenin to Rickstein's substance, biotransformation of Rickstein's substance to cortisol by Carvularia lunata, and then cortisol can be biotransformed to prednisolone by the organism Corynebacterium simplex, while cortisone derived chemically from cortisol can be biotransformed by Corynebacterium simplex to produce prednisone. So, here starting with sigmasterol to progesterone and using this Rhizopus nigricans, we get the 11 hydroxy progesterone which is converted to hydrocortisone, and then this with the help of Corynebacterium simplex can be converted to prednisolone. And also if we start from diosgenin, it is converted to Rickstein's substances, and with the help of Carvularia lunata, it is converted to hydrocortisone cortisol, from which cortisone is obtained, and on these the Corynebacterium simplex can act and produce prednisone. So, these are the steps involved in the combined chemical microbial transformation of steroids. Then cholesterol derivatives may be obtained from the biodegradation of cholesterol by enzymes produced by Mycobacterium species. Mycobacterium utilizes cholesterol as a carbon and energy source, breaking it down into simpler metabolites through a series of enzymatic reactions. These reactions typically start with the oxidation of the steroid ring structure or side chains by enzymes such as cholesterol oxidases. Mycobacterium species often possess multiple enzymes and pathways that act sequentially to break down the cholesterol molecule into metabolites like cholestforenin 3,1, pregnenolone and other intermediary products. In an industrial scale, large scale biotransformation use low cost sterols like diosgenin from Mexicum yam or stigmasterol from soybean oil. Microbial transformations employ cells, spores, enzymes or their immobilized forms. Active cultures grow 6 to 24 hours before adding substrate, or a large inoculum allows immediate substrate addition. Solubilizers like twin or solvent aid poorly soluble compounds. Sterilization, aeration and progress monitoring via chromatography or spectroscopy ensure the process ends at maximum titer. Let us now discuss about the application of live organisms as therapeutic agents. One of the important approach is the phage therapy. Phage therapy or phagotherapy involves using bacteriophages to treat bacterial infections. It emerged in the early 20th century, but was largely replaced by antibiotics after World War II. Some of the key advantages are reduced side effects, lower risk of bacterial resistance compared to antibiotics, high specificity sparing the host and beneficial bacteria like gut microbiota. Phage therapy also has a high therapeutic index, meaning it causes minimal side effects even at higher doses. How does this phage therapy works? Phage therapy works by using bacteriophages, viruses that target harmful bacteria. The process involves number one, bacteriophages attach to bacterial cells and inject their genetic material. In step 1, we can see that the genetic material produce new phages inside the bacteria. So, there is entry of the phage and degradation of the host DNA. Then degradation, the bacterial cell being destroyed as the new phages are released. The process halts the bacterial infection by preventing the bacteria from multiplying. We need to isolate a virus for this application. In a typical process of phage isolation, a bacterial sample is combined with an environmental sample from effluent outlets, sewage or even soil and allowed to incubate usually overnight. So, here we have the environmental sample with the phage and then we have the bacterial culture. We are culturing it by phase enrichment with the host culture. Then finally, we go for centrifugation, filtration and then we go for plaque formation assay and again the phage concentration to obtain the phage. After incubation, the remaining bacteria are separated from the culture using techniques like centrifugation or filtration and the resulting filtrate is assessed for the presence of phages. Then testing the isolated virus is very important. So, we go for plaque testing. These mixes dilutions of phage filtrate with bacteria applied to a plate via spreading or a soft agar overlay. Incubation reveals plaques indicating phage activity. Then we go for the culture lysis. Here phage filtrate is introduced into a broth culture containing bacteria and incubated. The culture is observed for cell lysis, which is indicated by the loss of culture turbidity. Alternatively, metabolic dyes can be employed to evaluate bacterial metabolic activity instead of turbidity. Let us now discuss about the screening of viruses. While a detailed biological characterization of a phage may seem useful for phage therapy, practical screening typically focuses on a few key characteristics like ability to eliminate target bacteria. Phages must be virulent and productive enough to outpace bacterial growth and prevent bacterial overgrowth. It should have strictly lytic behavior. Lytic phages are preferred in therapy because temperate phages may carry toxin genes which could increase the virulence of bacteria through genetic transfer. Host range, the phages that can infect a wider range of hosts within a specific pathogen species are more likely to be useful for treating infections caused by that pathogen. Phages with these ideal traits are stored in phage banks in bacterial medium. The mode of treatment and administration. Phages are highly specific to certain bacteria, often necessitating patient swabs and cultures before treatment. However, clinics maintain phage cocktails for common bacterial strains, periodically updating them to address emerging strains. In eastern countries, these cocktails are available in pharmacies. Phages are administered orally, topically on wounds or on surfaces. They are often freeze dried into pills with some types stable up to 55 degrees centigrade and a shelf life of about 14 months. Oral administration is more effective with an antacid to improve survival through the stomach, while topical application involves applying phages on gauze placed over the treatment site. Funding for phage therapy research and trials is challenging due to complexities in patenting bacteriophage products and the limited public awareness. Several trials have been conducted to evaluate phage therapy's efficacy against specific infections like Pseudomonas aeruginosa and Escherichia coli. Locus Biosciences has developed a CRISPR modified phage cocktail showing promise in early trials against E. coli urinary tract infections. The FDA has recently approved various clinical trials including intravenous and nebulized phage therapy for Pseudomonas aeruginosa infections in cystic fibrosis patients and trials for urinary tract infections and chronic prosthetic joint infections conducted by Adaptive Phage Therapeutics in collaboration with the Mayo Clinic. Phages in biofilm treatment. Phage therapy is highly effective against biofilm infections, particularly those caused by Pseudomonas aeruginosa and Staphylococcus aureus. In a report of 78 biofilm infection cases, 96% of patients showed clinical improvement, with 52% achieving complete relief or bacterial eradication. Challenges such as low antibiotics penetration due to structural barriers, enzymatic deactivation of antibiotics make phage therapy an attractive alternative for managing such infections. Here you can see in the first case there is attachment and then after that there is colonization, and there is growth, and there is maturation and dispersion taking place. So, these is basically showing the treatment of biofilms with bacteriophages. Another interesting application is in cancer. We have certain oncolytic viruses. An oncolytic virus refers to a virus that selectively targets and eradicates cancer cell. As it infects cancer cells, it induces oncolysis, leading to the destruction of these cells while producing new virus particles that contribute to further tumor elimination. Several viruses including adenovirus, reovirus, measles, herpes simplex, Newcastle disease virus and vaccinia have undergone clinical testing as oncolytic agents. While most oncolytic viruses are engineered for tumor selectivity, naturally occurring viruses like reovirus and Seneca virus have also been tested in clinical trials. The mechanism of action is the immunity as an obstacle. Oncolytic viruses selectively target tumor cells due to the latter's defective antiviral response. The interaction between oncolytic viruses and the immune system revolves around two key aspects. First one is the immunity as an obstacle. The patient's immune system poses a significant challenge to oncolytic viruses, as it naturally works to neutralize any virus present. To address pre-existing immunity, less common human pathogenic viruses can be used, yet subsequent antibody generation remains unavoidable. Strategies like coating viral vectors with polymers such as polyethylene glycol have been attempted to shield them from antibodies, although these also interferes with their interaction with host cells. Another approach involves hiding viruses within macrophages, exploiting their natural migration to areas of tissue destruction, particularly low oxygen regions associated with cancer growth. Here another mechanism of action is immunity as an ally. Despite impeding viral activity, the patient's immune system can also aid in fighting tumors. Infection caused by oncolytic viruses attracts the immune system's attention to the tumor, potentially generating beneficial and enduring anti-tumor immunity. Tumor lysis releases substances like tumor associated antigens and danger associated molecular patterns, triggering an anti-tumor immune response akin to a personalized cancer vaccine. So, here you can see the virus is infecting a tumor cell, and it is replicating within the tumor cell and resulting in lysis of the tumor cell and release of viruses, leaving behind certain debris, and then the virus goes on to attack other tumor cells. So, this debris will activate T cells and then lead to the T cell mediated tumor cell death. Naturally occurring oncolysis. Vaccinia virus has been studied for oncolysis since the 1920s, when Levaditi and Nicolau demonstrated its tumor inhibiting effect in mice and rats. Vesicular stomatitis virus shows promise for brain tumors, inducing regression and durable cures in animal models. Then reoviruses efficiently replicate in cancer cells, leading to lysis and forming the basis of reolysin. Then there is Seneca virus, an oncolytic picornavirus, is being explored for cancers like small cell lung cancer and pediatric solid tumors. There is Rigvir, an ECO7 ecovirus, is approved for cancer treatment in Latvia, Georgia and Armenia. Other viruses include Maraba virus, Coxsackie virus, then modified influenza A viruses and Sendai virus are under investigation for their oncolytic properties. Then we have the genetically engineered oncolytic viruses. These are specialized viruses that have been modified through genetic manipulation to target and attack cancer cells selectively while sparing healthy cells. These viruses are designed to exploit the defective pathways present in cancer cells, allowing them to replicate and lyse the malignant cells specifically. The genetic modification in these viruses often involve altering their viral genomes to enhance their tumor specific replication and destruction capabilities. These modifications can include tumor selective replication, enhanced tumor cell killing, immunomodulation, reduced virulence in healthy cells, and there are preclinical trials and clinical trials going on. Chen showed that CV706, a prostate specific adenovirus combined with radiotherapy enhanced cell death and viral burst size in prostate cancer mice. Then there is HSV 1716, safuravir demonstrated synergy with cancer chemotherapies, while bevacizumab, an anti-VEGF antibody, reduced inflammation and boosted oncolytic SSB virotherapy in mice. Then there are certain things in clinical trials. For example, Onyx 015 combined with chemotherapy showed improved responses, but was discontinued due to inconclusive results. Then Vaccinia virus GL-ONC1 was tested with chemo and radiotherapy in head and neck cancer patients as part of the standard of care. Then we have certain approved therapeutic agents like Telimogene, Laharparvec, known as T-VEC and developed by Amgen, successfully concluded phase 3 trials for advanced melanoma in March 2013. In October 2015, the US FDA granted approval to T-VEC, marketed as Imlygic, specifically for treating melanoma in patients with inoperable tumors. This marked its status as the first approved oncolytic agent in the Western world, leveraging the herpes simplex virus as its foundation. Now, certain things are in phase 1 trials for pancreatic cancer and phase 3 trial for head and neck cancer combined with cisplatin, chemotherapy and radiotherapy. Then there is Tesarpaturev, known as Delitect and developed by Daiichi Sankyo Stance as the inaugural approval oncolytic virus therapy sanctioned by the Japan Ministry of Health, Labor and Welfare. Delitect is a genetically modified oncolytic herpes simplex virus type 1, has gained approval for treating malignant glioma in Japan. Let us now discuss about other therapeutic agents, which include vaccines, nutrient supplements and probiotics. Microorganisms play a vital role in vaccine production and are employed to produce antigens or components that trigger the desired immune response. Bacteria, yeast and viruses are manipulated to express specific proteins or genetic material that mimic the antigens present in pathogens, aiding in the development of vaccines. With the aid of recombinant DNA technology, these microorganisms are harnessed to create safer and more effective vaccines, offering a controlled and reliable means of antigen production without the need to handle the actual disease causing pathogens. Then we have the food supplements like vitamins. Certain bacteria and fungi are capable of synthesizing vitamins like B vitamins like riboflavin, cobalamin and biotin are produced by microorganisms through fermentation processes, and also amino acids are produced by bacteria and yeast, and some of these include glutamic acid, lysine and others through fermentation. These are essential components in various food products, animal feed and in pharmaceuticals. Then another important application are the probiotics. These are live microorganisms that, when administered in adequate amounts, offer health benefits to the host. In top we can see yogurt and then kimchi. These are traditional food rich in probiotics, while bottom is the Yakult. It is arguably the most popular probiotics brand today. Then most common probiotics include strains of the beneficial bacteria such as Lactobacillus and Bifidobacterium, found in various fermented foods like yogurt, kefir, kimchi, sauerkraut and some type of cheese. These probiotics are known to promote a healthy balance of gut bacteria, aiding in digestion, immune system regulation, and potentially reducing the risk of certain conditions such as diarrhea, irritable bowel syndrome, and certain allergies. So, with this we come to end of this lecture. Thank you for your patient attention.